Okay, so here we are back again, and here is the review of proteins that I want you guys to know. I want you guys to remember, amino acids make up polypep or make up proteins, and they're linked together by peptide bonds. Okay, so here we have amino acids, and they're linked together by peptide bonds. There are 20 different amino acids that exist. Okay, and how these amino acids are actually linked together, and which of the different amino acids, how they're actually linked together, along with which amino acid is where, and the sequence of them makes up the protein. Okay, so um, how these amino acids are linked together, meaning which amino acids located where, and how many amino acids are there, and the location of them determine which protein that will be. And there are several different proteins in your body that function differently. You have different enzymes in your body, and depending upon how those amino acids are linked together will make up that protein, will determine what enzyme that is. Um, Another important part is I said that they are linked together by peptide bonds. A lot of you probably remember from doing macromolecules, macromolecule web quests, that sometimes proteins are referred to as polypeptide chains. And polypeptide chains are actually just a chain of amino acids. And what does poly mean? Many. What does peptide mean? It's referring to the peptide bond, so it's referring to many peptide bonds. Also, another little bit of review is peptide bonds actually are what is detected by Bio-Rad's reagent. If we go back to back to the macromolecule labs that you all did, Bio-Rad's reagent detected proteins, and it wasn't actually detecting the amino acids, but the peptide bonds that link the amino acids together. Moving on. So there are several different functions of proteins. There's 7 of them: storage, transport, regulatory, movement, structural, enzymes, and defense. We have all these different proteins in our body functioning together, and they make up most of our body. We have transport, where we're transporting oxygen through red blood cells. We have regulatory hormones that regulate our bodies. We have movement, so we have muscles that help with movement. We have structural, which are what you learned our cell membranes are made up of. Our hair, nails, bones are made up of proteins. Then we have enzymes that speed up the rate of chemical reactions by lowering the activation energy. And then we have defense, which are antibodies. They're our immune system. So here we have RNA, and what you guys should have learned before is RNA has 3 differences from DNA. 2 of those differences are RNA has a sugar called ribose, and it also has a nitrogen base, uracil. Okay, just like deoxyribose nucleic acid DNA, we have ribose nucleic acid now. Because of the sugar, it's changed. And we also have a phosphate group. Now remember, guys, adenine, the 4 bases that we talked about before, the 4 nitrogen bases, were adenine, thymine, guanine, and cytosine. Now we have adenine, uracil, guanine, and cytosine. Adenine is going to bond with uracil, and just like in DNA, guanine and cytosine are going to bind together. Okay, so the function of RNA. The function is to obtain information from DNA and synthesize it into proteins. Okay, so we have DNA in the nucleus, and it actually contains the genetic information, the genes that we have. It's actually instructions to build proteins. It's like a recipe for proteins. The different sequences of nucleotides, nitrogen bases found in DNA, actually code for amino acids. And we'll go through that process. And what helps us is RNA. It actually obtains the information from DNA and helps us synthesize proteins, make proteins. So here we have three differences from DNA. We have one, it is single-stranded instead of double-stranded. So RNA actually is a single-stranded organism, while DNA is a double-stranded organism. Or not organism, molecule. Whoops. An organism is a living thing. DNA is not a living thing. It has ribose. It has a ribose sugar instead of deoxyribose. And it contains uracil instead of thymine. There are three types of RNA. We have messenger RNA, mRNA, which copies information from DNA for protein synthesis. And actually, what this mRNA does is we have DNA in the nucleus, and DNA is actually never going to leave the nucleus. Think of the nucleus as this protective safe for DNA, okay? And it's going to stay in that nucleus. It's never going to leave. So what does it do? It actually makes a copy of itself in messenger RNA. So it makes a copy of messenger RNA, which is just a copy. It's like a photocopy of DNA. And that photocopy is actually going to go out into the cytoplasm. Now, on messenger RNA, we have something called a codon. And this is 3 base pairs that code for a single amino acid. Now, if you'll remember, the base pairs that we find in messenger are the base pairs or nitrogen bases or nucleotides that we find in RNA are adenine, uracil, guanine, and cytosine. If we take 3 of those base pairs on the RNA, each one of those is going to code for an amino acid. Now here it's pointing out 3 of the base pairs, or 3 of the nitrogen bases, or 3 of the amino acids, or I'm sorry, 3 of the nucleotides, and that 3 is going to code for an amino acid. Now, for an example, A, U, G will be 3 nucleotides, and that's actually what starts out every single protein synthesis, or every single protein. Starts off with AUG, that's our start codon, and AUG actually codes for MET. We have another one called transfer RNA. And transfer RNA actually collects amino acids for protein synthesis. On the bottom, you'll find an anticodon, a sequence of 3 bases that are complementary base pairs to the codon in the mRNA. And based on the amino acid that it's carrying, it will have the anticodon that matches with the codon. So for example, I said AUG codes for MET. On the anticodon, we'll have the exact, the complementary base pairing on that. So AUG would be down here on the codon. We would have UAC. So we would have UAC as an anticodon, and that's just going to come and bind to the codon, and it's going to bring with it that amino acid that I said the AUG codes for, and that's MET. We also have ribosomal RNA, and this is the easiest one to remember, rRNA, because just like it says, ribosomal, all it is a ribosome, okay? And ribosomal RNA actually is just RNA that combines with proteins to form ribosomes, okay? So all that is just a fancy word for ribosomes. And it's just RNA wrapped around proteins to form a ribosome. And this is actually going to be the site of protein synthesis. This is actually where protein synthesis is going to occur. Think of this like a workbench. Like if you were building, a birdhouse, you would have a workbench. Well, a ribosome is going to be your workbench. Amino acids. Amino acids are the building blocks of proteins again, and there's at least one kind of tRNA present for each of the 20 amino acids used in protein synthesis. So we'll have at least one type of tRNA that has that anticodon down at the bottom, bringing with it an amino acid. And two ways you can remember tRNA. tRNA transfers amino acids. So if you can remember that T stands for transfer, then hopefully you can remember it transfers amino acids. And what that means is it actually takes amino acids to the ribosome. And we will tie all this together in just a minute. You can also remember it because it kind of looks like a T, does it not? What do you think the term protein synthesis refers to? Well, what do you think? If you remember back, protein, or I told you that synthesis actually refers to making, okay? So to make something. And proteins is, a protein, so to make proteins. That's actually what protein synthesis means, is to make proteins. And protein synthesis actually occurs in two steps. The first step is called transcription, and the second step is called translation, okay? So mRNA is made from DNA and goes to the ribosome, okay? So we have mRNA, it's made in from DNA, and it goes to the ribosome, okay? Then we have translation, and this is where proteins are made from the message on the mRNA, okay? So here we have DNA, and it's going to stay in the nucleus, okay? Remember I told you that DNA is located inside the nucleus. The nucleus holds the DNA. And think of it like a protective safe, okay? The nucleus is like a protective safe to DNA. And DNA are the blueprints or the recipes for proteins, okay? So the cell wants to keep that very safe. So it locks it in its nucleus. And it's not going to let those instructions or proteins leave. So you know what it does? It says DNA, it says messenger RNA out. So it'll send messenger RNA out with the actual message of DNA. So mRNA will actually bind, and we'll talk about this in just a minute, will actually form a complementary strand of DNA to one side of the DNA, and it'll go out to the ribosome where proteins are made. Transcription: in order for cells to make proteins, the DNA code must be transcribed to mRNA. And I actually don't like this representation of it because it makes it look like we take DNA and we just put it into the exact same copy of mRNA. And actually, mRNA will be a complementary base sequence. So A will bind with U, T will bind with A, G will bind with C, C will bind with G. Moving on. The mRNA carries the code from the nucleus to the ribosome. Now don't forget that, because the nucleus contains DNA. The DNA will never leave, and the ribosomes are actually located out into the cytoplasm. So we need a messenger to take the instructions from the DNA out to the cytoplasm, to that ribosome where that protein is actually made.